So we'll go ahead and get started. So it's a pleasure to introduce Matt Rockman, who's faculty at New York University, NYU. Matt's got his PhD from Duke in 2004. And Matt is an evolutionary geneticist, and I think does a really beautiful set of integrative work on understanding the causes and consequences of genetic variation in a number of different systems to answer a number of broad questions about the maintenance of variation. So Matt works, as I say, across a number of different systems, including polychaetes and nematode worms. Today, he's going to be talking about his work in nematodes. He's visiting our lab at the moment, and so he'll be around for the next month or so. So if folks are interested in having coffee with him, by all means, email him or myself, and we can set that up. With that, take it away, Matt.
Great, thank you. Yeah, I'm here every afternoon for the entire month of May, and grad students, postdocs, I will buy the coffee. So I want to hear about your research. I would love to meet and talk to you more. Today, I want to just introduce you to some of the work we're doing on Caenorhabditis nematodes. And to kind of motivate this, I will tell you that I recently read this book, which is a biography of Haldane, who's a very charismatic, dynamic character. I think on the page, he's really fun. In real life, he would have been insufferable. But it's a really interesting story about his career in genetics. But the thing that really caught my attention is this discussion about a kind of disagreement between two of his last students, John Maynard Smith and Helen Spurway. And they disagreed about how to pick a system for research. And the basic distinction is that Maynard Smith thinks you should start with your question and then find the organism that is best for that question. And Spurway argued that you should start with an organism, get to know it, let the organism tell you what the questions are. So so I started with questions. I started with questions about basically, is there something about the genetic basis for phenotypic variation in animal development that influences the way animal development evolves? That's kind of the motivated question. And so question first, then organism. And so I picked the organism that is easiest for asking questions about the genetics of development. And that is, I think, without a question, and apologies to the Drosophilists, C. elegans. They have a 3-day generation time. They can cross or self, and we can cryopreserve them. They're just so easy. And so over many years, we've developed a lot of different tools for studying genetic variation in populations and how that genetic variation influences phenotypic variation. And I'll just sort of name some of the resources we've developed so that you know they exist. One is that we have now, in collaboration with the Teotonio Lab in Paris, about 1,000 resequenced recombinant inbred lines from a 16-parent cross that has passed through more than 200 generations of evolution. And so these things are so highly recombinant that we can pretty trivially map stuff to individual variants because there's just so many breakpoints in this population. We also have a panel of 800 and some near isogenic lines that tile a single 1.5 megabase region on one chromosome. So 800 breakpoints tiling across that little tiny region there that allow us to, again, to detect really subtle effects of these very small intervals. More recently, we've developed a big panel using a genetic trick in C. elegans of mitonuclear swaps. So here I'm showing you six nuclear backgrounds and 6 mitochondrial backgrounds, and we have this 36 panel of every possible nuclear and mitochondrial combination. And actually, we now have an 18 by 18 version of this. And so we can look at very subtle effects of mitonuclear interactions. So the point of all of this is that we can ask these questions using this worm because it's so easy, and so we can develop resources quickly. But having now spent a lot of time with these worms, crossing them, looking at them under the microscope, and living worm life, I'm now pretty much totally sold on the Helen Spurway model. It turns out worms are really delightful. And when you get to know them and love them, then it stops being a question of worms, can you tell me about quantitative genetics? And more an issue of like the worms being just over here, got something cool for you to look at. And I follow the worms now. So the worms are setting the agenda. And today I want to tell you about 2 evolutionary genetics ideas that we came to because the worms told us to go there. So I'm going to give you 2 pretty unrelated vignettes. There's a common theme, but it's stuff that the worms told us was going to be interesting. So we're going to start with a question about why worms have the sexes that they do. In most of the species of Caenorhabditis nematode have males and females. C. elegans, our famous model, has males and hermaphrodites. But I want to start with other species of Caenorhabditis, most species, which have these separate male and female sexes. And so here's a schematic showing you male and a female. The males are a little bit smaller, and they have just the one sex chromosome, one copy of the X, no Y chromosome. So it's an XO male, and the females are XX. And this means that sex determination in this system is chromosomal, and basically it's governed by the sperm that fertilizes the X-bearing oocyte. So we expect that half of the sperm will have an X and half of the sperm won't, and the progeny will be 50-50 male and female. And so for totally unrelated reasons, we were studying the reproductive schedule of a bunch of worms of one of these male-female species called Caenorhabditis BCI. And here's what we observed. We do a cross between a male and a female, and we throw the male away, and we track all of the progeny that the female produces. And what we found is that the earliest progeny that that female lays are disproportionately female, 2 females for every 1 male. And over time, as the female continues to have offspring from this cross, it reaches a 50-50 sex ratio. And late in that female's life, she's laying mostly males. So there's this time course of offspring sex. And this implies that there's something about the X and the not X-bearing sperm that's different, that causes fertilization to favor the X-bearing sperm. And so if we kind of glance inside the reproductive tract of a female nematode, they have 2 gonad arms that go one anteriorly and posteriorly from the vulva. And here we're just looking at a little piece where there's oocytes developing over here, and they're going to be ovulated into the spermatheca, where there are sperm hanging out, and then one of them will fertilize that egg, which will then be passed into the uterus to develop. So you see in the uterus, there are developing embryos undergoing cleavage. The sperm get there by being transferred during a mating by the male. They sorry, I should point out that the sperm are amoeboid. I realized that I showed you a little graphic of sperm and egg, and if you're not used to thinking about worms, you would think, why is there like a Pac-Man ghost? But that's because the sperm don't have tails like we're used to seeing. They have, they're amoeboid, they crawl with pseudopodia. So here, the sperm are transferred into the uterus, and then they crawl into the spermatheca. And in that spot, they're then competing, jockeying for space and for fertilization opportunities. And each time an oocyte is passed through this spermatheca and into the uterus, it's pushing some sperm out, and then they have to crawl back in. And so there's a lot of work that each sperm has to do in order to maintain its position. And they stay there to the extent that they can through several days of egg laying. And so somehow the sperm that have the X and that don't have an X are different, and in a way that makes the X-bearing sperm more competitive, or possibly just different in a way that allows the female to distinguish them. And it could be that the female is choosing the X-bearing sperm rather than the X and the O being differentially competitive. At any rate, that difference has to be there between the 2 kinds of sperm. And that can't really be due to transcription of genes on the X. It's not really about the X chromosome genes, because that's all transcriptionally silent when the sperm are being generated. So it really has to be about cytoplasmic stuff segregating differentially, or membrane stuff. Stuff that's not about the genotype of the sperm itself, but about the spermatocyte that divides to make the sperm. That stuff gets segregated, and some of it goes with the X and some of it doesn't, and that's what makes the difference. So when we think about the genetic causes of this difference, we don't have to be thinking about X chromosome genes. We can be thinking about autosomal genes that contribute to the pattern of cytoplasmic segregation. So that got us thinking about why, before we get to that, let me just mention that I showed you this time course of development that starts with females, female offspring. If there's a remating, that female mates another male, then she's resupplied with a pool of sperm, and then the brood stays disproportionately female indefinitely. If the female dies before using up all of her sperm, then her brood is disproportionately female biased. And so if we just let a population go, we end up with a female biased population. That's kind of an equilibrium condition for these worms. So even though it looks like a time course, and ultimately it's 50-50 if we look over the course of a single brood, in a population in more natural conditions, it's going to end up with a female biased sex ratio. So we wondered, why would these worms have a female biased sex ratio? And that got us thinking more about the natural history of these animals. And so we've been studying Caenorhabditis nematodes out in the wild for a long time. And in particular, we've worked a lot in Panama. This is a former graduate student, Saul Sloat, doing some field work in Barra, Colorado Island, which is popularly known as BCI, which is the name we gave to the worm species we found there, CBCI. And Saul here is looking for worms. And when we find the worms, they live in rotting stuff. They eat bacteria. All of the Caenorhabditis species eat bacteria. And so when we find them, they're typically in a little patch of rotting stuff. This is a Spondius mombin fruit that has rotted at that spot. And that's going to be just totally full of worms. And the worms had to get there somehow. They can't actually persist or proliferate in soil or in litter or anything that's not full of wet bacteria. Is not suitable habitat for the worms. So they have to get to that patch somehow. And then they're going to go through some proliferation with that food resource. And then that resource is going to run out of food, or it's going to, in some other way, stop being appropriate habitat for these worms. And then they're going to need to move on to the next patch. And they can't just crawl across this landscape because it's dry and just otherwise not suitable. And so what typically happens is that when they've exhausted a resource, then they will get environmental cues that induce development into a dower larva, which is a larva. It's pre-reproductive, and it's got morphological adaptations that facilitate migration. And in particular, they're able to hitch a ride on some passing animal. So they're basically phoretic on beetles or flies or snails or some other animal that's going to pass along and drag them from one rotting patch to a new one. And so the result is that there's migration to a patch, proliferation on that patch, and then migration, some number of individuals to a new patch. And that kind of patchy landscape is exactly the kind of population biology condition that gives rise to female biased sex ratios. So this is expected from the literature back from Hamilton's work decades ago. The basic idea is that if we have lots of patches that are founded by a small number of individuals, and on each patch, the population is going to grow until it runs out of resources, and then its final generation is going to represent migrants that can go to a new patch. If we have, in this case, just a two-generation patch founded by a single mating pair, then they're going to have some number of descendants that's governed by basically the brood size of the females. That's kind of the limiting factor in how big this population can grow. If we have a mutation that causes a female biased sex ratio, each of the females now can have its own brood, and that rarer male can mate with all of those females. So the population will grow bigger in the same period of time, have more migrants as a result of that two-generation cycle. And so variation among patches allows patches that have female biased sex ratios to have more migrants from one cycle to the next. And that favors the evolution of female biased sex ratios. Of course, at the same time, there's issues going on within patches. So I showed you a single mating pair as the founders. If we have a patch that has a bunch of founders, then they're all going to have their offspring. If there's now variation within this patch in the sex ratio, then we're going to end up in this G1 generation with a different number of males and females. And here the males are rarer. That means that each of those males is going to have more offspring on average than each of those females. That's the kind of classic Fisherian sex ratio logic, that in a panmictic population, the rarer sex has an advantage. And so through frequency dependence, we end up with a 50-50 sex ratio. So we've got these two kinds of phenomena going on here, competition within patches and competition among patches. And we also have in these nematodes a kind of unusual system of sex determination, which is chromosomal sex determination, where in principle, the autosomes could be doing all the work, and where we know for sure that the sperm made by the males are playing a role. Most theory about sex ratio evolution involves maternal control of sex ratios. In particular, there's so much work about haplodiploid systems where the females, in a lot of cases, really can be just deciding on the fly. Here we have a case where the paternal genotype we know has to matter. The maternal genotype may also matter, but the paternal genotype does for sure. And so Saul, this graduate student who was working on this project, really wanted to know, does it matter which parent is controlling the sex ratio? And it turns out that there's surprisingly little attention to this question in the literature, and most folks who have mentioned it have said, well, there's no relatedness asymmetry between autosomal genes and males and females. And so autosomal genes coming from the mother and coming from the father should experience the same degree of relatedness to each of their offspring independent of sex. There should be no force that makes those things different. And Saul wanted to test that. And so he used simulations. He simulated a whole bunch of patches, and he seeds them with some small number of worms. From our work, we've estimated very indirectly that the number of founders of a patch is typically on the order of 3 to 5 individuals. So it's a pretty small number per patch that starts it out. So he's going to seed a patch with a number of individuals. All these patches get that number of individuals. Then there'll be some number of generations of proliferation within that patch. And then he'll extract a sample of the whole field of animals to represent the migrant pool, and then he'll get rid of everybody and start the cycle again. And so it's exactly the phenomenon we think is happening in our worms. And then this goes through many cycles of this colonization, proliferation, and extinction. So now we can talk about the genetics of the system. Saul modeled a worm typical genome. And so the model we're going to look at is not a single locus kind of model. It's a thing where there's a constant influx of new mutations and the population is evolving in response to that. And so he's going to allow for 4 different kinds of mutations. There's mutations that act in males to increase the male frequency or decrease the male frequency. There's mutations that act in females in the same way. And then he's going to compare how evolution plays out if sex ratio is governed totally by the mom or totally by the dad or by both of them simultaneously. And the big surprise at first was that indeed, the equilibrium sex ratio is different in these 3 cases. So this is, I think, I strongly believe not a claim that is present in the literature. And it was surprising to us, and we did lots of things to make sure that we could write the model in different ways and get the same result. That the maternal case gives the result that's described in the literature, but if the paternal genotype governs things, then we get a more female biased sex ratio. And if they're both affecting the sex ratio, and in our model it's totally additive, then we get a kind of intermediate value of the sex bias. Sex bias evolves in all cases. That's just a function of this patchy population structure. And indeed, as we add more founders per patch, so here, this is now founder number on this axis and the equilibrium value of the sex ratio on the Y, you'll see that more founders results in something that gets closer and closer to 50-50, which is just what we expect because of this kind of competition between the within and the among patch levels of selection here. So that all looks right. And so, so we have this thing where the paternal effects are acting differently from the maternal effects. And so this raises the question of what's going on in this case where both parents are playing a role. Is it that they are sharing an optimum under this circumstance, and so they're both evolving to this shared phenotype, or are they evolving antagonistically, where the males are pulling it to a more female bias sex ratio and the females are trying to push it back up? And we can just look at the effects of the alleles that have spread through the population in the model. And what we find is that indeed, this is sexual antagonism. It's runaway sexual antagonism. So every time a male, a paternally acting female biasing allele appears, it spreads. And every time a maternal male biasing allele arises, it spreads. And so these things are going to pull in opposite directions indefinitely, as long as there's the input of mutations that are in these directions. And this results in this kind of intermediate equilibrium actual sex ratio that is the balance of these things. At the kind of molecular evolution level, this should cause lots of genes to be experiencing positive selection, even though there's no phenotypic readout from this phenomenon at all. Because they're in the same population and they're balanced. So that was a surprise as well. And so the question that remains kind of is like, what's different about the paternal effect? Why would that matter in this case? And the basic punchline is that the selection here is really dependent on the variation among patches. That's what's facilitating the selection of the patches that grow fastest and that have the more biased sex ratios. So variance among patches is the key thing here. That implies that there might be differences in the among patch variation for the maternal effects versus the paternal effects. And we can measure that among patch variation using a statistic QST, which is just the genetic variance among patches, basically. I'm describing this, by the way, in a kind of a group selection language, because it's the most intuitive way for me, but you can rewrite all of this stuff in terms of correlations within patches in a kind of more Hamiltonian way. It's all the same. So the punchline here is that indeed, when we look at just maternal or just paternal, the variance among patches is higher when we have paternal regulation of these things. And when we look at the genetics of the fathers in their loci that are affecting sex ratio, the variation among patches is greater than in the maternal case. And if we look at this across different population sizes, here we've got QST on one axis and the sex ratio equilibrium on the y-axis, and these dots represent different numbers of founders. What you'll see here is that the 10 founder paternal effect case has the same ratio, the same sex ratio as like the 6 founder maternal effect case. And that's because they have the same QST. The among patch variance is the same in those two cases. And so you get the same sex ratio. When you get really small numbers of founders, like 2 down here, things get different because now a lot of your patches are founded by 2 individuals of the same sex. And as the sex ratio bias gets more extreme, that becomes more common. And so there's a kind of a limit on how far that can go. And so this is the case of maternal only or paternal only. When they're evolving in the same population, now each of these pairs has to be at the same sex ratio because they're in the same population. But again, the QST for the paternally acting loci is higher than QST for the maternally acting loci. And so basically what's happening here is that each batch of founders on a patch is getting males and females. But as soon as that patchiness induces female biased sex ratios, then the number of founders per patch is greater for females than for males. And that results in greater among patch variance just through sampling effects. You're sampling fewer males per patch than you're sampling females. That results in greater variance. So it's the most trivial and kind of, in retrospect, obvious explanation here that female biased sex ratios result in fewer males per patch and greater variance per patch. So this is all based on a kind of worm-specific population biology scenario. And so Saul wanted to know if this kind of generalizes. And it turns out that it basically doesn't matter how we think about the population biology as long as we've got patches. So we've got our mating after colonization case in the nematodes, but in other versions of this, including a kind of classic local mate competition models where you've got your mates all in the same, your mating animals, you're mating before dispersal, like with figs, where the mated figs fly out and found new patches, the mated wasps in the figs, thank you. You get the same result. In all of these cases, if you have simultaneous paternal and maternal regulation, then you get runaway antagonism. And if you have just male or just female, then you get different equilibrium sex ratios as a function of the different variance among patches. And there's one model where this doesn't work, and that's kind of the one generation mating before dispersal, which is actually the kind of canonical local mate competition model. And so it's possible that the reason nobody has observed this before is that that canonical model actually doesn't have the right circumstances for this variance to differ between the males and females. But any of these conditions where you've got multiple generations going within a patch, regardless of whether the mating's before or after, we always get the same result here. So It doesn't matter how many generations per patch, more generations actually amplifies the differences. Doesn't matter discrete or overlapping, except very subtle quantitatively. And so the punchline of this first vignette that the worms led us to is that sex ratio is generically different in all patchy systems when the regulation of sex is under maternal versus paternal control, and that if it's both, then we get runaway antagonism. This is just due to sampling differences for the males and females once we've got a female biased sex ratio, and it's a very general result. So this is a case where I never imagined that I would be working on sex ratio evolution, but the worms told us that we should, and I think we learned something here. So I'm now going to switch gears, tell you a second vignette that is about our selfing species like C. elegans. So in our selfers, of course, the sex ratio is nothing like 50-50. It's pretty much all hermaphrodites all the time. Males are pretty rare. But that's useful for a lot of things. It means we can self to make inbred lines. And so we do all sorts of quantitative genetics where we get wild strains of hermaphroditic species. They're inbred in nature. So we just get these inbred lines out of nature. We cross them and then we self them to make recombinant inbred lines. And every time we have this kind of same result, which is that when we look at the frequency of the two founder genotypes across the genome, it's usually 50-50. Most of the time, that's what we expect. We put in equal amounts of those founders at the start. We cross them, we derive inbred lines, 50-50. But there's always a couple of spots in the genome where things are not 50-50, where we have really strong segregation distortion. And so here you see, in this cross between strains N2 and CB4856, the N2 allele on the left side of chromosome 1 goes almost to fixation. And we're not doing anything that we can perceive as selection in the process of doing this. That's this is a result from my postdoc in C. elegans. More recently, we did this with C. tropicalis, another of these selfing species. And here we've got two different regions of the genome that are behaving really weirdly. And it turns out that after lots of genetic analysis of this sort of situation, it's always due to the same exact phenomenon, which is, surprisingly, paternal affected gene drive. Parental affect gene drive, rather. And so the typical case is something called a medea, which is a maternal affected dominant embryonic arrest. And this is a very clever name that other folks gave to it. That's a figure from Greek mythology who murders her children. And that's what a medea element does. And you might think medea element, that sounds like a mariner element or some kind of transposable something. And so I want to emphasize that medea is just like a mode of inheritance. Right? It's any locus that has this maternal affect dominant embryonic arrest inheritance is a medea. And we've, as a community, characterized a bunch of them. They're all totally genetically unrelated things. And they're regular kinds of genes that just happen to also have this kind of drive result. And so to explain the kind of genetics here, let's think about a Punnett square where we've got two heterozygotes. Each of them has a medea allele and a wild type allele at the same locus. And they're going to make sperm and eggs. And the medea allele acts in the mother to make a toxin. This is a kind of schematic model that turns out to be literally correct for a couple of the cases that have been characterized. But in some way, there's something that's toxic to embryonic development that the mother loads into the eggs. All of the eggs get that. It's not a gene, it's a gene product. And so everybody gets that toxin. And the genes then segregate in the normal Mendelian way. But those embryos that inherit the medea allele genetically can then post-zygotically transcribe an antidote. And so they're all rescued. And the 1/4 here of the progeny that didn't get that M allele, they're dead. And so that's basically how this works, is there's a single locus that makes a parental effect toxin, and then later a zygotic effect antidote. And that's a medea. We also have paternal effect versions of this. This is actually the one for the N2 CB4856 cross in C. elegans. That's a paternal effect version of a medea that we named Peel. And so here the toxin is loaded into the sperm. And again, the zygotes that don't inherit that locus then die. So this was first discovered as a genetic phenomenon in flower beetles in 1992. And the basic result here is that we have an interaction between maternal and zygotic genotypes at a single locus, such that heterozygous mothers will produce dead offspring that are homozygous for one of the alleles, for the wild type allele here. And this results in an increase in the frequency of that Medea allele as the embryos that don't carry it are killed. And this turns out to be a pretty useful gene drive system. And as I mentioned, it's not a particular kind of gene. Any kind of gene that has this property will work. And so folks have engineered this into Drosophila, for example. And so it's just, you just need a maternal toxin and a zygotic antidote, and you can have a Medea element. So we, again, discovered this by accident in C. elegans. Then we discovered it by accident again in C. tropicalis. And at the same time, another group discovered other ones also in C. tropicalis. And in all of these, well, in many of these cases, we didn't just discover a Medea element here and a Medea element there. We actually found a Medea element here and another Medea element also right here. Both alleles at a locus have this Medea activity. And so then you've got an antagonism where one allele of the maternal heterozygote kills one class of homozygotes and the other allele kills the other class of homozygotes. And only the heterozygotes from that het cross will survive. And here's some real data. For an example, this is from this cross of C. tropicalis, just looking at a single locus, the results of a single locus segregating on chromosome 5 here. What we get is that the M1 allele kills the M2 homozygotes, the M2 allele kills the M1 homozygotes, only the heterozygotes survive in this case. And so that's kind of a striking phenomenon that makes us think, well, maybe that should result in some kind of balancing selection. And that then connects up with this other result that when we look at the sequences of populations at these loci, they're always hyperdivergent. The two haplotypes, the Medea and the other one, which sometimes is also Medea, they're always super divergent at the sequence level. So this is from C. elegans, where Zeal and Peel constitute the two molecular genes that make up this one Peel element. And the adjacent genes that have nothing to do with this interaction are super divergent. So at the protein level, these other genes that are not part of it are more than 5% sequence different at the protein level. The C. elegans genome has very little variation generally. So this is a huge divergence for protein sequences in this case. And if we look at it in various other ways, this is true for lots of regions of the genome in all three species of selfing Caenorhabditis. So in Briggsii and elegans and tropicalis, there's very little genetic diversity through most of the genome. And then there's little patches that are really hyperdiverse. And so in the C. tropicalis cross that I was just telling you about, we have these little tiny regions where if you align the two haplotypes, they're up to sometimes more than 10% divergent on a background of basically no divergence at all through most of the genome. So little islands of hyperdivergence. And all of the known Medea and Peel elements occur on those islands of hyperdivergence. So we've got that there's a lot of these Medeas and Peels, sometimes at the same locus. They always occur on divergent haplotypes. And there are lots of these divergent haplotypes spread all over the genomes of all three of these species, which raises the question of, are these Medeas and Peels causing that hyperdivergence? Well, we saw that if there's antagonistic Medeas and we do a cross of two hets, then only the hets survive. That looks like it should maintain diversity at that locus. Maybe it's balancing selection. And when we first found this, we did some quick simulations and we didn't see balancing selection. And so today I want to show you an analytical model that asks what happens in this kind of case. And so I'm just going to walk through very briefly to give the flavor of it. We're going to track the frequencies of the three genotypes at a locus where there's two Medeas segregating. So there's M1, there's M2, that makes for three genotypes. Each of them has a killing penetrance. And then also there's going to be some selfing rate. So in this model, we're going to allow offspring to arise from selfing with some probability and from outcrossing with the complement of that probability. So that's S and 1 minus S. And so now you can just write down what's going to happen in this population. So the probability of an M1 cross, selfing rather, is the probability that there's selfing times the frequency of that genotype. All of the progeny of that are going to be M1 homozygotes. So that's all we're doing here is just writing down what's the genotype of all offspring from all of the crosses. And so that's from the selfing. From the outcrossing, we have to have reciprocal crosses because it's a parental effect. So we need to know who's the mother and who's the father in a cross for each pair of genotypes. And so you just write all this down, and then you can write recursion equations that are going to say what's going to happen to the genotype frequencies from generation to generation. And then we can analyze the dynamics and find equilibria and so forth. So one really reassuring thing is that if there's a single medea that's not antagonized, it just sweeps to fixation. And any amount of outcrossing will allow it to sweep. If there's selfing, it's going to take longer because only the heterozygotes are experiencing killing. So if it's mostly selfing, then it takes a long time to get started, but eventually it sweeps to fixation. That's sort of obvious. It's worth noting that in simulations, we can see that, that with selfing, high rates of selfing, selection is really ineffective, right? Heterozygotes are rare, so at high rates of selfing, here we're introducing a medea at frequency 5% and asking about its frequency 100 generations later. And in blue, you'll see the high selfing rate situations where mostly it gets lost. In a deterministic system, it's always going to spread, but selection is just very ineffective when there's lots of selfing in this sort of situation. So that's all sort of obvious. But the fun part is that antagonistic medeas exhibit positive frequency dependence. And so the common one wins out over the rare one, even if the rare one is more penetrant. And so here we're looking at the case where the starting generation here at 20% is the more penetrant medea. It's going to kill 90% of the affected offspring. The resident medea has a penetrance of only 50%. And so under obligate outcrossing, if selfing is 0, the more penetrant thing always sweeps to fixation, no problem at all. But with a little bit of selfing, things change. And with a lot of selfing, then we get the opposite result, where it's deterministically eliminated from the population, even though it's more penetrant. And so we can analyze the recursion equations and ask what's going on. And basically, if we look for the equilibria for this system with antagonistic medeas, there's stable equilibria at a frequency of 1 and at 0. But then there's an unstable equilibrium at this horrible looking thing. What this shows is that the only thing that matters for that equilibrium is the selfing rate and the ratio of the penetrances. That's pretty straightforward. And the kind of punchline of that unstable equilibrium is that an invading medea has to be above that equilibrium frequency in order to successfully invade. If it's below that, then it's going to be deterministically eliminated, even if it's more penetrant. And so what that looks like is shown here, where we can imagine a perfectly penetrant medea that's going to kill 100% of the affected offspring, invading a population where the resident medea has the penetrance shown on the right. So this line is representing a resident that has a 5% penetrance. And what this shows is that when the selfing rate is high, then that totally penetrant medea has to have a frequency of above whatever this is, 3 or 4% in order to invade. That really weak medea can hold off that invasion under this positive frequency dependence. And even if at these other levels, you'll see that it's really difficult at high selfing rates for a rare medea to invade, basically, at all. I should say that in C. elegans and C. tropicalis, most folks have estimated the selfing rate to be much higher than 99%. So we're really thinking about the far right edge of this graph. The model I've shown you so far is kind of a simplified monoecious thing where every individual can self or outcross. That's not really how the worms work. And so it turns out when we put the real worm genetics in here, just Androdiaceae, then things are a little bit more complicated. And in that case, actually, peels and medeas behave differently because under Androdiaceae, males are always the result of a cross. So they have higher heterozygosity on average than the hermaphrodites. And so things are a little bit different, but the qualitative results are the same. So here's where we started is, are these medeas causing these divergent haplotypes? Well, the original hypothesis that folks have put in the literature is that maybe this results in overdominance. And so the killing of the homozygotes allows both alleles to persist for a long time and you get divergence. We show that that's not what's happening here. So it just doesn't do that in a population with selfing. So the alternative is that as we've shown, these things produce positive frequency dependence, which makes it difficult for an allele from one population to invade a different population that already has a medea. And so one possibility is that that's going to basically be little islands of isolation that can't flow back and forth between populations while the rest of the genome can. So if we imagine a typical locus in two populations, then the alleles can move back and forth freely. If these guys are fixed for different medea alleles, those medeas can't move back and forth because the number of migrants is always going to be a small fraction of the local population. And so they're sort of trapped locally while the rest of the genome is homogenized. And so that's one explanation that this now requires a certain kind of population structure for worms, which we don't know if they have. But this kind of produces some kind of testable scenarios for whether this could explain the distribution of medeas and hyperdivergent haplotypes in these nematodes. So that's a possibility. But there's another possibility that I haven't mentioned, which is actually the one that we proposed initially in 2008, that I'm kind of partial to, that just inverts the causality. So the way we started was maybe the medeas are causing the hyperdivergence. And the alternative is that hyperdivergence is causing the medeas. And that's the argument that I'm most attracted to. And it goes like this. Worms live in different kinds of environments, and they're adapted to those environments. And it turns out that in C. elegans, at least, the hyperdivergent haplotypes are enriched for genes that are putatively associated with environmental stuff. So there's a lot of receptors for different kinds of environmental cues. There's a lot of genes that have signatures of genotype environment association in those haplotypes. So it's possible that those hyperdivergent things are actually just regular old-fashioned ecological balancing selection loci that are old because the environmental conditions are also old. And so that's kind of just a normal balancing selection. In that case, we get these haplotypes that are really old, and that's plenty of time for medea type things to evolve accidentally. They have a lot of different genetic variation on them. Some of that genetic variation could be simultaneously parental effect toxic and zygotic effect antidote. And so in that case, the drive wouldn't have really anything to do with that. And one reason that I like this is that as we've seen, the medeas and peels only act in heterozygotes. But heterozygotes are vanishingly rare in all of these species. These species really are evolving almost all of the time as inbred lines, the whole genotype competing with whole genotypes of other individuals. And so these guys only act when they're heterozygous. Heterozygosity is super rare. And so the way that we could think about this is that this is a kind of like backwards sheltered load. So sheltered load is more typically thought of as this situation where you have recessive deleterious stuff that is never exposed to homozygosity, and so it can just persist in highly heterozygous regions. So if you have very strong balancing selection at MHC, for example, recessive deleterious stuff can accumulate there because homozygotes for that locus are going to be vanishingly rare. And so a thing that only acts when homozygous is invisible. And this is the flip side. This is a thing that only acts when heterozygous, now present in things that are selfing all the time. And so that's an alternative hypothesis here, is that it's hyperdivergence first for regular ecology reasons and medeas and peels second, just because these haplotypes are old. So we have a lot of work to do to figure out if there's data in support of any of these models. But we're also left with this big mystery, which is why are we finding so many of these things in Caenorhabditis when basically aside from the beetles, they're not present anywhere else. And I feel like Drosophists would have noticed these if they were there. So I think there's something special and weird about worms, and I don't know what it is that's special and weird about them. That's a question I would love to get feedback on. I want to thank the folks in my lab, in particular Saul, this guy here, who did the sex ratio model. And I would be delighted to take some of your questions right now, and also for all of May, just to remind you.
the second part, you said that depends on a particular model population structure for C. elegans. Don't know how population structure works in C. elegans? I mean, you had a lot of details in the other species.
Right. Yeah, I think we don't know how population structure works in C. elegans. So in particular, the thing that's required for the model that I described is that local populations are fixed for a medea, and different populations are fixed for different medeas. And I think at the moment, we don't really have enough of a sense of what's a local population to know whether the distribution of those medea alleles is consistent with them being kind of fixed locally. And
Do you have the same sort of metapopulation dynamics? C. elegans?
yeah, so at the very local level of rotting patch, proliferation thin, migration to a new patch, yes.
In the first part of the maternal biasing, were you simulating the same mutation rate? Is there the same type of genetic architecture for maternal and paternal biasing? Does that
Yes.
influence things?
Right,
Is it any
so,
simpler in the paternal side than maternal side?
right, absolutely. So, yeah, in this case, there's influx of mutations all the time. It's kind of a typical worm mutation rate, actually, that's here, but it's equal rates for the maternal and the paternal components. And so in real biology, there may be different mutational targets for those two things, very likely, and that's not in this model at all.
Would that, do you think, change the dynamics or
Yeah, I bet it
compensate
would.
for one of the variables?
Maybe, yeah, and I think, yeah, so what would it do? It would, if it affects the relative variance of QST, then for sure it should affect, and it certainly could. There's also a thing that's not in the model at all, which is that if you think about mechanistically what's going on here, it's like the X and the non-X sperm are becoming more different, and the females are being maybe better able to distinguish them and choose between them. And maybe there's a limit on how different the sperm can get that comes earlier than the ability of a female to distinguish between them. So there's lots of ways of thinking about what's actually going on here inside the animal. And so we've made a bunch of kinds of tools for doing the quantitative genetics of this trait. We've got CBCI worms that fluoresce green or red, depending on sex. And we've got big plans to do lots of worm sorter experiments to generate the kind of sex ratio data that we need on a really large scale. And we have a panel of about 400 CBCI recombinant red lines. And we can do maternal and paternal separately to look at genetic variation on one component or the other by using a reference strain. So getting to the molecular stuff in CBCI, that's in the works. But the basic point is that the model is ignoring lots of important biology.
So I apologize if I'm understanding incorrectly, but it seems like with the the N1, that system, selfing is more beneficial since fewer offsprings die.
Yep.
In that case, is there any kind of sexual preference or chemical cue that they're using to choose mates that are like them in a sense?
Right, yeah, so there could be, we don't know of anything like that right now, but it's absolutely right that sorted mating should reduce the problem that comes from these medea elements. Selfing should reduce the problems. And so the way we originally, this is our paper on the C. tropicalis system, the empirical part, not the model. Sorry, let me just put it up here. Our paper is the one on the left where we hypothesized that selfing is actually an avoidance mechanism, and that it's a selective force, that the medeas are a selective force that favors high rates of selfing in these androdioecious populations.
I know Graham left someone in charge of ending the question session. Or is it me? Okay. I was kind of like waiting for.
Yeah, I have a question also. So in the last part, do you find divergent islands without any ideas, or
Yeah.
like the opposite?
Yeah, so there's tons of those hyperdivergent islands, and most of them have no evidence for medea. the thing that comes out of the model is that really weak medias are actually really potent. So a media that's just a few % penetrant, and it doesn't even have to kill the offspring, it just has to slow their development. And we know that some of the medias do that, they just slow larval development a little bit. And that's the kind of thing that if you're not looking for it, you're never going to see it. So I think it's super possible that there are just tons of really low penetrance medias everywhere. I think that's biologically plausible. The one kind of test of plausibility is that a medea element has to be expressed in the maternal germline. And a PL has to be expressed in the paternal germline. And so I looked at all of the islands of hyperdivergence and asked, are there genes like that? And for the current annotations in C. elegans, 86% of the hyperdivergent islands have the right kind of germline expression, and the remaining ones don't, which could be that we're not looking at the right thing. So I think that that's kind of ambiguous evidence. But yeah, so it would have to be lots of really weak stuff that we wouldn't notice unless we tried really hard to find it. And because people rarely study heterozygotes, I think it's easy for them to have been missed.
Any more questions?
Thanks.